Good morning, everyone. Firstly, I think it's a huge privilege for me to stand here and talk to you about this topic because most of the research and the background to this comes from this institute. It was so nice to meet a lot of people whose publications have contributed to a lot of inspiration to this work. And I also have the privilege, as Professor Ghoshal said, to have been trained in Mumbai and then done all my postgraduate training in Australia. So essentially had the best of both worlds. This is a topic of my PhD, which I'm currently pursuing. And it would be great to hear what your views would be. As I said, that a lot of inspiration for this is from this institute and the work done by the people here. How will this work? All the way down. All right, so I'm going to talk very briefly about the role of microbiome in health and disease. What are the currently established diagnostic techniques? What is the study that we have done in Brisbane to utilize molecular techniques to assess bacterial load? We have some preliminary results which I'm looking forward to share with you all. And what would be our next steps? So firstly, a very brief overview of the gut microbiome in health. Why do we talk about the gut microbiome? I think there are two main important reasons. It plays a very important role in metabolism, and the other area is protection. When it comes to metabolism, it is the main organ that is responsible for energy production by the fermentation of a lot of non-digestible substances. It is essentially responsible for synthesis of a lot of vitamins. Next is protection. We know that the intact mucosal lining of the gastrointestinal tract prevents the invasion of pathogenic organisms. It certainly has an antimicrobial effect and is responsible for stimulating the local mucosal immune system and also the systemic immune response. Talking very briefly about the gut biogeography. So different regions in the gastrointestinal tract have different physiological functions and clearly hence different composition of the microbiome. The small intestine in particular has the role of acting as a site of absorption, plays a very important role in immunity, and also has the sensory function. The surface area of the small intestine is essentially 10 times larger than the large bowel, which is quite amazing. This is just a diagrammatic presentation of how the biliary tree enters into the small intestine, and it will be very relevant to what we talk subsequently. I've mentioned there that there is a very low bacterial load in the small intestine, and that is something I'd like to concentrate in my next few slides. So firstly, why do I say that there is a very low bacterial load in the small intestine? Nature has got a number of protective mechanisms that prevent bacterial colonization. Most importantly is the gastric acid, which lowers the pH and prevents bacterial growth in the stomach and the proximal small bowel. Next are the enzymes, which are the very harsh proteolytic and lipolytic enzymes. Then is the bile, which again prevents growth of bacteria. There's a very rapid antegrade movement of the luminal contents from the proximal bowel towards the ileum. There are other systems which I've highlighted, and one of the most important ones is an intact anatomical ileocecal valve, which will prevent the reflux of the colonic contents into the small intestine. So these essentially are nature's mechanisms in place to prevent bacterial overgrowth. Hence, as we go from top to bottom, the laser doesn't point properly. From the stomach down, we can clearly see that the stomach and the proximal small bowel has got a very low bacterial load of 10 raised to 1 to 10 raised to 3 colony forming units per mil. And as you move towards the terminal ileum or the distal small bowel, the bacterial counts are as high as 10 raised to 7 colony forming units per mil. And the colon is densely populated with gram-negative and anaerobic bacteria, with the counts being as high as 10 raised to 12 colony forming units per mil. So we clearly see that there is a transition between the upper and the lower gut, and that is not just in the amount of or the density of bacteria, but it is also in the type of bacteria. So how would you characterize the gut microbiome? I could simplify it for myself in three ways. One is, what is the composition or the type of bacteria which colonize the gut? Secondly, what is the density of this bacteria? And lastly, what are the metabolic products or the byproducts produced by these gut bacteria? We've spoken about the gut microbiome in health. Now let's talk about what happens in a diseased condition. I would like to now introduce you to the topic of gut microbial dysbiosis. In simple terms, I could define it as alterations in the composition, density, and the function of the intestinal or the gut microbes. It is essentially an imbalance between the good or the beneficial microbes and the harmful or the pathogenic microbes. Why is this important? We now know, looking at this tree, that gut microbial dysbiosis not only plays an important role in the pathogenesis of a lot of gastrointestinal conditions, which could be inflammatory bowel disease, irritable bowel syndrome, bowel cancer, but also in a lot of non-gastrointestinal conditions, which could be rheumatoid arthritis, metabolic syndrome, diabetes, and so on. So essentially, characterization of the gut microbiome is going to be very critical to understand the pathophysiology of various gastrointestinal and extraintestinal disorders. Next, I'm going to talk about small intestinal bacterial overgrowth, or SIBO, as an example of gut microbial dysbiosis. This is a relatively new term that is becoming more and more relevant in the literature. SIBO has been defined as the presence of excessive and or an abnormal type of bacteria in the small bowel. This can be different type of bacteria. Traditionally, it was restricted to only colonic type of bacteria, but now we also include oropharyngeal type of bacteria. There is clearly a lack of consensus in this area with regards to precise definition for SIBO. There is no consensus on this. There is a lack of diagnostic criteria, and the gold standard for the diagnosis of SIBO is largely debated as well. We'll discuss these more in detail in the subsequent slides. So historically, when SIBO was defined, it was defined as by aspiration of jejunal fluid and culture, with the counts of bacteria being more than 10 raised to 5 colony-forming units per mil. However, these aspirates were taken from patients who had a post-surgical anatomy or stagnant loop syndrome, and clearly this has thought to be a very, very high cutoff criteria. SIBO is now thought to be very relevant to functional gastrointestinal disorders as well, and in these conditions, the counts of the bacteria are not as high as those seen in post-surgical anatomical conditions. So the recent cutoff criteria is 10 raised to 3 colony-forming units per mil. But again, this is felt by some experts to be very, very high. So there is a significant debate about what should be the diagnostic cutoff value for SIBO, which puts a huge question on the validity of the definition itself. Let's talk very briefly about the established diagnostic techniques. So firstly, what is available? In clinical setting, the most common tests available are breath tests. I think this is very familiar to this institute because a lot of work in breath test has been done here, as we've discussed with Professor Ghoshal on many instances, and as Prof highlighted as well. Direct tests are essentially proximal small bowel or proximal jejunal aspirate and culture. But in clinical practice, at least from where in Australia, I've never seen this being done, though interestingly it is done here. So essentially I thought it was a research setting test, but clinically, at least in India, we now know that this is done. So now we've been focusing on molecular techniques, which are culture-independent methods, which include measuring the bacterial density by utilizing the quantitative PCR or doing a sequencing to see the microbial community profiling. We'll talk very briefly about the breath test, which I essentially measure two gases, hydrogen and methane. So these have been developed to overcome the challenges and the limitations of culture and aspirate. They are very widely available. They can be done very easily. They are quite cheap and they are very efficient in an outpatient setting as well. You don't require any specialized training or any very expensive expertise or instruments. What's the principle of the breath test is human cells are not capable of producing hydrogen and methane. So the presence of these gases indicates that bacteria are fermenting the non-digestible carbohydrates. The breath test is done in the following manner. You ingest a substrate like glucose or lactulose. When this reaches your proximal small bowel, if there are bacteria present as a result of overgrowth, they will be fermented and hydrogen and methane gases are produced. These then get absorbed into the systemic circulation and will be exhaled by the lungs. You then measure the concentration of these gases at baseline and at various intervals, and this is done by gas chromatography. You get a graph which is depicted in the diagram there, and this is the cutoff criteria currently used for diagnosing SIBO. Rise over baseline in the hydrogen by 20 parts per million and or methane by 10 parts per million is defined as a positive breath test for diagnosing small intestinal bacterial overgrowth. These are clearly not without limitations. The most important ones are there's a high rate of false positive and false negative results. False positive results can occur in situations which are associated with carbohydrate malabsorption, for example, chronic pancreatitis, celiac disease. And false negative can occur in conditions which can affect the intestinal motility, for example, gastroparesis. There is again a lack of universally accepted substrate and cutoff values for defining a positive result. Many centers now use glucose, but in not so long ago, lactulose, which is considered as a marker of colonic transit, was used for doing breath tests, and that has given rise to a lot of false positive results. The sensitivity and the specificity is very, very poor. So glucose breath test, which is more commonly used, has a sensitivity of 60% and the specificity is about 80%. So clearly, the most commonly used breath tests are not without limitations. Now we talk very briefly about the direct test or the currently accepted gold standard. Geogenal fluid aspirate is the gold standard for establishing the diagnosis of bacterial overgrowth. How do you get it? We can't use a normal gastroscope because it will not reach beyond the fourth part of the duodenum, and the aspirates have to be obtained from proximal duodenum. So you use a specialized gastroscope which is called as an enteroscope. This fluid is then obtained and it is cultured. But the big problem here is, as Professor Ghoshal said, only about 70 to 80% of the microbial stains are not cultured. That is, only 20% or 30% at the most can be cultured by the routinely available culture methods. So that is a huge limitation that we have. In addition to that, there is lack of diagnostic cutoff values, which we've already discussed. And there's lack of standardization as to which site would be better. Is it the proximal duodenum? Is it the proximal duodenum? There is still an ongoing debate about that as well. Similarly, the false positive and false negative results can be high as well. So clearly, there is, I'm just going to be ahead a little bit, sorry. A lot of limitations to direct and indirect tests. In this setting and against this background, we thought of developing and validating molecular techniques which can be used in clinical setting. The main aim here was to find and precisely quantitate the bacterial density and also to characterize the bacteria which colonize the mucosa-associated microbiome. So now coming to the question, which I think Professor Holtman has briefly highlighted, which is better? Is it the stool microbiome or is it the mucosa-associated microbiome? He's already discussed this very much in detail, and I just want to re-emphasize the point that the stool microbiome is transient. It is clearly affected by what you eat. But the mucosa-associated microbiome is like your fingerprint, and hence all the studies that we are doing and plan to do will be essentially concentrated on doing on the mucosa-associated microbiome. How do you do this? Do an endoscopy, get a biopsy. This biopsy specimen is then frozen in RNA later, and it is stored at negative 80 degrees centigrade. Next steps would be to extract the genomic DNA. Once this DNA has been extracted, it is used essentially for two purposes currently. One is amplify the 16S gene, and then it can be sent off for sequencing. Or the next thing that we're doing is we're measuring the bacterial density. The study that we've done, and I'm going to talk about, is essentially concentrated at determining the mucosa-associated bacterial density in biopsies obtained from the second part of the duodenum. We selected 3 patient groups. The first group was asymptomatic people. These are patients who were referred to our hospital for bowel cancer screening, who had no symptoms and only had a positive fecal occult blood test. Or patients who had iron deficiency anemia but no gastrointestinal source for this and had negative endoscopy and colonoscopy findings. The second group was those with functional gastrointestinal disorders. We had patients with functional dyspepsia, irritable bowel syndrome, and a huge number of patients with an overlap of functional dyspepsia and IBS. And the last cohort was patients with inflammatory bowel disease, both with ulcerative colitis and Crohn's disease. So these were our 3 study groups. We did an endoscopy on them and again used our Brisbane aseptic biopsy forceps to obtain the biopsies. So this forceps, as Prof. Holtman has explained, works in a slightly different way, that the biopsies can be obtained aseptically. It is introduced in the similar fashion as the normal forceps is introduced through the working channel of the gastroscope. Once it enters the duodenum, the sterile plastic cap is sheathed off, and the biopsies that you thus obtain are completely aseptic. So this is the process that we used in our study here. Next is after obtaining the biopsies, we extracted the DNA and amplified the 16S gene. Then we used the quantitative PCR to obtain the bacterial density, which was expressed as a ratio of 16S RNA to human beta-actin. We did this by using the standard curve method. To check our aseptic biopsy forceps against the routine biopsy forceps, we randomly selected some patients. These patients had biopsies taken by 2 techniques. One is using the special forceps and one is using the standard biopsy forceps. The aim here was to compare which of the collection method is better. Is it actually better or not? These are our preliminary results, and I'm very happy to share them with you all here. So far what we have seen, so far we have managed to recruit 283 patients who have had their endoscopic assessment done. 170 of them were females. Their mean age was 50 years. 70 patients were control patients, or those who had no endoscopic findings and no symptoms. We had 94 patients with functional gastrointestinal disorders. 69 had an overlap of functional dyspepsia and IBS. 22 had only functional dyspepsia, and interestingly, we had only 2 patients just with irritable bowel syndrome. So once again, highlighting the fact that overlap is more common than what we anticipate. We had 111 patients with inflammatory bowel disease, 69 with ulcerative colitis, and 42 with Crohn's disease. And we had 9 patients with organic diseases like gastroesophageal reflux disease, Barrett's, and so on. Now we look at the results of bacterial density in the different groups. So we observed that the bacterial density, which was expressed as a ratio of 16S to beta-actin, was highest in the functional group. That was 0.2. This was significantly higher and almost twice the number for the asymptomatic controls at 0.1. The bacterial density was lowest in the patients with inflammatory bowel disease at 0.02 and 0.01. And all these differences were statistically significant. So essentially, we were very intrigued that those with functional gastrointestinal disorders have got the highest bacterial load in their biopsies obtained from the second part of the duodenum. And those with inflammatory bowel disease had the lowest bacterial load. We then looked at the effect of proton pump inhibitor I'm sure we remember from the previous slides, we said acid secreted by the stomach prevents the growth of bacteria. So we wanted to see what happens in patients who are on a proton pump inhibitor. If you are on a PPI, irrespective of which group or cohort you belong to, the bacterial density was significantly higher as compared to those who are not on a PPI. There's a huge difference, as you can see there. The PPI users had a bacterial density of 0.2, whereas the non-users had a bacterial density of 0.05. So the message here is clearly proton pump inhibitor, which inhibits the acid secretion, has got a negative impact in protecting the colonization of the bacteria in the proximal small bowel. Next is we looked at the comparison between the devices. So we took samples from 98 patients and we found that there was no correlation if you took the biopsies from the standard forceps and the aseptic devices. So essentially the standard forceps did not bring the results that we saw with our aseptic biopsy forceps. So the conclusions of the study so far is the bacterial densities in patients who've got functional gastrointestinal disorder is significantly higher as compared to asymptomatic patients or inflammatory bowel disease patients. The next point here was proton pump inhibitor treatment is definitely associated with higher bacterial loads, and this is not explained by the difference in study cohorts. And lastly it's an important study point. If you have a high bacterial load, it could possibly be a predictor to response of antibacterial therapy in patients with functional gastrointestinal disorders. So we've got a long way to go. This is just the first few initial steps. As I said, we've done the bacterial density, but we also want to know if there is a signature microbial organism which could predict these differences. Hence, we've now looked into the sequencing and are working on this area. This is something that we are waiting the results for eagerly to become available to us. The next things are, I've spoken about, we want to do, we are in the stages of doing the sequencing. The other test that we're doing is the breath test. We're also measuring the intestinal permeability in these patients and assessing their sensory function by how they respond to a nutrient challenge and what symptoms they produce. And the routine clinical information is also being collected. So this will still take some time, but I'm really looking forward to this study and what results will come through from here. So thank you very much for all your attention.